Morning, Dr.
Hello,
Slade.
people. Hello, Raphael. Welcome to
I
day
have learned
2,567 of the COVID isolation period. I hope you're all happy. I see the usual cast of characters here. Thank you for coming.
I have learned it is much harder to get Roman Legionnaire armor than I thought it would be. It's also hard to join one than I thought it would be, considering they don't exist anymore. There goes that retirement plan.
Well, let's see. 4 times 4 is 16. So we've got 15 people here out of close to 50. Let's wait a couple minutes, see if anybody else shows. Or maybe I'll just give everyone who showed up on time an A for the class. What do you say, guys? Thumbs up?
Sounds good.
Elena's in. Sydney wants an A. Heaven wants an A. Edgerman wants an A. Oh, a lot of people want A's. Good, good. Well, unfortunately, it doesn't work that way. You need to earn your A. How many people know someone who's died of COVID at this point? Give me a thumbs up if you do. No, give me a thumbs down if you know someone who died.
I don't know how to give a thumbs down.
Isn't there a thumbs down? I thought there was a thumbs down. I know someone who died today of COVID, an uncle of mine. Directions. Oh, there's thumbs up or clap. Okay.
My grandmother's neighbor passed away of COVID.
My uncle, who used to live in Vestal, New York, died today. He was over 90. But, oh, thank you for your sorrow there, Hebron. Okay, shall we rock and roll? It's 5 after. All right, we are recording this time. The last couple of times I haven't recorded, which was just an accident, but it's the way it worked out. So I didn't have any recording to post up. Chat. I'm bringing up my chat. Bringing up my visages. Someone just came. Hello, whoever you are. Okay, shall we rock? Let's do it. Okay, cancer cells. What are cancer cells? They are cells that are basically growing and growing and growing without any regard for any external cue to divide. So like we've been talking about, most cells don't divide unless they're specifically stimulated to divide. Cancer cells are just internally stimulated to divide. They're self-sufficient in terms of that. And there are a lot of other changes in cancer cells that we'll get into. So just some big broad definitions here. There's 3 different types of tumors. One are benign, and benign's when it's kind of all contained within itself, so the tumor doesn't move anywhere. They often keep getting bigger and bigger, but they're not getting any, not spreading. So here on slide 3, you can see there's almost like a capsule around it. Actually, it is a capsule. It's a labeled capsule. So it's not sneaking out of there. It's all staying there. So that's benign. And benign tumors usually aren't that bad. Sometimes people don't even take them off. If you've got a dog, a lot of times dogs will have fatty tumors. They just leave them because it's not worth taking them off. They don't cause any problems. A malignant tumor then is one that starts to invade. And this is where the name cancer comes from, because when they look at the tumors, they look like they've got these claws sticking out like a crab. And those claws are like little sections of cells that are invading neighboring territory. And that's when things get dangerous because, for one thing, they start to invade into tissues and destroy tissues, and that can cause damage. But more importantly, once something gets malignant, it's just a couple steps away from becoming the next step, which is metastatic. And metastatic, here you can see there's this basal lamina you see here. We'll talk about that quite a bit. When cells cross that basal lamina, that's when they're malignant. And the problem with that is, like, the basal lamina is between your epithelium and the inside of your body. So your skin layer has got a thick protein layer between it and what's actually inside the rest of your body. And that's a really nice barrier there. But these cancer cells will find a way to get through that. And once they do, they can invade other tissues. And they can also go into a blood vessel or a lymph vessel. Once they get in there, they travel along with the flow, and then eventually they'll slip back out of that. And when they do, they set up new tumors. So metastatic is when they're really dangerous because they come out and set up tumors in other areas. So, like I always say, if you've got breast cancer, not that big a deal. It's just a breast, So that won't kill you if you've got a bad breast. But the problem with breast cancer is it goes metastatic. And then it goes to the lungs, it goes to the liver, it goes to all different places and disrupts function in important organs. And that's how the cancer usually kills you. So typically we worry about malignant because it can go metastatic, and you want to get rid of it while it's still just malignant, because metastatic is when cancer is really dangerous. Okay, so you got your benign, your malignant, and your metastatic. And I throw these out here just to show you why usually when things go metastatic, a lot of times where they go is in the lungs or in the lymph nodes. And that's because, if say you've got, again, let's just go with breast cancer. You've got breast cancer, these cells get into the lymph, and from the lymph they go, the next place they get is the lymph node. So you've got cells that come in here, they get in the lymph node and they get stuck there and they set up tumors in the lymph nodes. If they're in the blood, then if you imagine something goes into the bloodstream from a tissue, what's the next capillary bed it sees? From the tissue, you're in capillaries, it gets into venules, and then into veins, and then into the heart, then it goes to the lungs, and hello, here's your next capillary bed. So that's why a lot of things will metastasize to the lungs or the liver. If it's coming from the intestines, the next capillary bed would be the liver. And those are important organs, so that's why they end up there. Okay, so what are the differences between normal cells and cancer cells? One thing that's different is they tend to be de-differentiated. So we talked about differentiation in the little section on Xenopus embryos. Early embryo cells are very generic, pluripotent, multipotent, totipotent. They can just become just about anything. And that's kind of their role, is to divide and make more cells and then differentiate into something else. So embryonic cells are dividing and they're not performing any function. So here you've got your hematopoietic stem cell. It doesn't carry blood, it doesn't do any immune function. All it does is just divide and divide and divide. So once it divides, it can then differentiate into a B cell. Now it's got a function. Now it's doing something. But B cells don't divide. They're done dividing. So typically, when you look at cells and cell division, cells are either dividing or they're fully differentiated, and they're post-mitotic. They'll never divide again. So when you get cancer, a lymph-type cancer, say you get these cells here, lymphoid. Can everybody see this? Have I done this correctly? I hope so. Get this lymphoid progenitor cell that's dividing and dividing and dividing. Well, it's not performing a function. So your blood gets filled up with these non-functional cells. And that's why that cancer is really bad. So de-differentiation. One change in cancer cells is they become more embryonic in character, which means they divide more and they're not performing any specific function. And another thing, Loss of communication between cells and the environment. Okay, so this is kind of important. We talked a little bit about this. Oh, wait. We're still doing differentiation here in this slide. Let me get the slide 10. Okay, so here's what normal tissue looks like. Here's your basal lamina, which is a thick layer of protein. Here is the underlying tissue, the rest of your body. Here are your skin stem cells, so they're dividing. And as they get, like one of these cells will divide and make two, one of the cells goes up and starts to differentiate. And as it gets closer to the surface, it differentiates more and more and more. And differentiation in the case of these squamous cells means the cytoplasm gets filled up with proteins, and it makes a protein barrier. And eventually, the cells die, and they're just dead protein barriers at the top of the skin. So that's normal progression of differentiation. When cells become cancerous, okay, when cells become cancerous, so these stem cells keep dividing and dividing, and they're not becoming skin cells anymore. They're just dividing cells, and this would be your tumor here. And you can see it's erupted, it's broken through a barrier. When it comes through this basal lamina, that's when it's metastatic. That's when it's malignant. Okay, so that's one changes in the cells. They become more embryonic and less differentiated. Number two, this is a terrible figure, by the way. I would read the notes on this because I couldn't find a half-decent figure that explained what I wanted to say. But the next one is it loses its cell-cell contact information. So loss of communication between the cells. Two different things here. One is connection with the focal adhesions to the extracellular matrix, the collagen and fibronectin and stuff like that. So we talked about this all the way back in chapter 14 when we were talking about cell crawling and these focal, what am I saying? Focal adhesions? Yeah, focal adhesions are grabbing onto the extracellular matrix and pulling the cell forward with it. So that's one thing the focal adhesions do, but mainly they're connecting the cell to the extracellular matrix. And there's two-way communication going here. I talked about this in the last class in terms of trophic factors. The cell needs to be connected to that extracellular matrix so it knows it's in its right contact. And when it lets go, when it no longer has that, then it goes into apoptosis. So most cells need that. That's called anchorage-dependent growth. And then also, the other type of connections are the adherens junctions. And these are like cell-cell connections, so not cell extracellular matrix, but cell-cell connections. And this is also producing signal transduction. And what these do is when a tissue is done, it's full, it's got as many cells as it wants, these cell-cell connections tell the cells to stop dividing. So two things going on here. One, if it lets go, it dies. And if it's in a nice close contact with other cells, it stops dividing. So cancer cells will lose both of those. So even though they're in a tissue, they'll keep dividing anyway, and that's how you get a tumor. It's just cells piling up on top of cells, and they're in the way. And also, once they slip out of that tumor, they no longer have this anchorage dependency, so they can survive without being stuck. Okay, why does the heart not have cancer? I believe, heart cells are post-mitotic, so they're typically not dividing. We'll talk in the next class about what cells are most likely to become cancerous. Epithelial cells are your most dangerous ones, and that's because they're exposed to the environment and they get a lot of toxins and a lot of DNA damage from that. Muscle cells tend not to be that, tend not to get cancerous. Sometimes a skeletal muscle or bone will get a cancer, but mostly it's epithelial cells. 90%, I think, of all cancers are epithelial. But, and again, heart cells are not normally dividing, so they're, so many more steps away from becoming cancerous. Epithelial cells, you've got that stem cell layer, they can be induced to become cancerous a lot easier because they're closer to the division. Okay, so what the hell is I talking about? Okay. So we've got two things going on here. We've got anchorage dependent growth and what do I call the second one? Density dependent inhibition. So those are your two things, anchorage dependent growth, density dependent inhibition. So it doesn't divide when it's in a tissue and it dies when it gets out of that tissue. So that's something that cancer cells lose. So they can do both. All right. The next thing is telomerase. Telomerase. Oops. What is this?
Sorry, I didn't get that.
Go away. Okay. Telomerase. So telomerase is based on the fact that eukaryotes have got linear chromosomes. And whenever you make replicate the DNA, you have to start every new strand with a piece of RNA. So you get an RNA primer and then DNA takes off from there. Then the cell has to come back and take that RNA out and fix it. Problem is, when you get to the end of the chromosome, this very end, you've got an RNA primer. You take that RNA primer off, now there's nowhere to start the strand to finish it off, because DNA can't just start from nowhere. It has to start from a free 3' end. Yes, 3' end. So when you replicate this DNA, you lose a little bit of information. And every round of replication, you get another gap at the end. So your chromosome gets shorter and shorter and shorter. So every time the cell divides, it gets shorter, shorter, shorter. And they realized this early on when they started doing cell culture, because they could culture cells and they get like 50 passages of cells, which means 50 cell divisions, and then all the cells would die all at once. So the cell needs a way to protect its chromosome so it's not getting shorter and shorter every time. And that's what telomerase does. Telomerase is a protein, it's an enzyme with a RNA template as part of it. So what it does is base pair with the end of the DNA, and then now the DNA's got a template that you can feed off of. And so it base pairs with the DNA and then finishes out that strand. Add your bases. And now it leaves like the same thing behind that was there before. So the enzyme can kind of like drop down and come back up, and now it's base paired again. So it's kind of like adds 6 bases, 6 bases, 6 bases. It just keeps adding repeats onto the end of the chromosome. So it extends that 3' end, and then you can just do the second strand, and then off you go. So it's protecting the end. Now, if that doesn't make sense, watch this video. This is a GIF animation I made like in the 90s. Very annoying making this. It was basically draw a picture, take a GIF, move something, take a GIF, move something, take a GIF, move something, take a GIF. It was just like forever. But I think it explains things pretty well. You can go ahead and look at this. I actually don't have access to this anymore. The only way I ever get it is by using the Wayback Machine to find it. But take a look at that. I think it's a good animation. So that was it, telomerase. So another thing that cancer cells have is they've got active telomerase that's protecting the chromosome, and that effectively makes them immortal. They're not going to die after a certain number of divisions. Anybody have questions about that? I went that kind of fast. Well, happy? Okay. Okay, self-sufficient growth signals. This is probably the most obvious difference in cancer cells. Basically, like I said earlier today, most cells need to be stimulated with a growth factor to be stimulated to divide. These cells have got that those pathways activated without the growth factor. So we'll talk about this in the next class, but basically, if you look at that receptor tyrosine kinase pathway, any of those enzymes along the way, if they were mutated to become constitutively active, you wouldn't need the growth factor anymore. So everything downstream just happens if you've got one molecule that's activated. So RAS is a big one. RAS is mutated in a lot of cancers. So basically, the RAS mutation changes it so it can't break down GTP anymore. So normally it breaks it down, goes to GDP, and picks up GTP and shuts it off. And this RAS picks up GTP and it stays active forever. So now these cells are constantly dividing because everything from RAS down gets activated. So self-sufficient growth signals. They don't need to be stimulated to divide. Angiogenesis. Angie, she was my neighbor when I was growing up. That's not true. That is not true. Angiogenesis. I'm just trying to arrange myself better here. Okay, angiogenesis. Angio is blood vessel, genesis, making new. So this is making new blood vessels. And next slide. This is a nice animation. Oops, a nice animation too. I'm not going to play it for you now, but you should watch it when you get a chance. Basically, excuse me, a tumor can only get so big before its size is limited by the ability of oxygen to diffuse in. In your body, every cell is within, I believe, 3 cell widths of a capillary. And that's because that's about as far as oxygen can go efficiently. I mean, it'll go everywhere, just slower and slower as you get further and further away. So every cell is within 3 cell widths of a capillary. Well, as this tumor grows and grows and grows, the center of it gets further and further away from a capillary. So you end up with a necrotic center in these tumors. So a tumor will get to be about 1 to 2 millimeters in diameter before the center starts to die. So then when cells divide and it gets bigger, the center dies and it collapses. So it just kind of, it's stuck at that size until it starts to get its own blood supply. And it does this by releasing growth factors. It will release VEGF, vascular endothelial growth factor, and it will release FGF, fibroblast growth factor. And these factors will stimulate the capillaries, and the capillaries are made up of single endothelial cells. So these growth factors will hit those endothelial cells, they'll start to divide and move, and then they'll form new capillary networks that will invade that tumor, and now the tumor's got a blood supply. And then it can get as big as it wants. And also at that point, it's got an escape valve. So now once it becomes metastatic, it can go into the blood supply and off it goes. So angiogenesis is really important in terms of cancer growth. And there are a lot of different strategies now to get rid of, to block angiogenesis to make tumors shrink. Okay, is that it? I guess that's it. That's what I got. Okay, so those are the ways that cancer cells are different. Let me just see if I can't make this bigger. Okay, the way that cancer cells are different from other cells, they're dedifferentiated, they're more embryonic. They lose communication with neighboring cells. They have telomerase active, so they can be immortal. They have their own growth signals. They don't need the body to tell them to grow. And they bring in their own blood supply. Now it's like another organism inside you, basically. It's got everything it needs. Okay, hallmarks of cancer. What else is there we need to know about cancer here? One of the hallmarks of cancer is invasion and metastasis. As I said, it's not really considered cancer until it starts invading neighboring tissues. So how does it do that? Well, first thing it has to do is stop being connected to other cells. The cell is connected to extracellular matrix with its focal adhesions. It's got adherens junctions that's connecting cell to cell. So these things are, I think I talked about this last time too, either that or the other class, developmental biology, I talk about it. They're transmembrane proteins that are connecting to extracellular proteins and cytoskeleton on the inside. And on the inside, while they're connected to cytoskeleton, they're activating signal transduction. So this is communication in both directions. So the cell knows that it's connected to something by activating signal transduction, and signal transduction can tell it to let go, grab or let go. So one thing cancer cells do is they'll let go. They'll let go of their with their cadherins, which is your adherens junctions, and let go of their focal adhesions. And now they're free. Now they can start crawling. So the first thing they need to do is let go of other cells, and that has to do with these connections. Did I just get chatted? I'm sorry. I've got this tiny little screen here, and I'm having a hard time moving things around. I believe heart cells do not divide. Okay, so no new chats here. Okay. Okay, letting go, loss of cell-cell connections. The other thing they have to do is start moving. How do cells move? Well, hello, chapter 14. We talked about chapter 14 way back when. I think that was last year. Seems so long ago now, doesn't it? So chapter 14 was talking about cell crawling, and that has to do with hormones activating receptor tyrosine kinases, which then activate guanine nucleotide release proteins, which activate G proteins. And the G proteins important in moving were RAC-RO CEC42. They make the stress fibers, the filopodia, the lamellipodia, and that's what's all important for division. So once the cell's got its self-sufficient growth signals, the growth signals are the same as the crawl signals. So they activate that whole process, and then they can start crawling around. So invasion and metastasis, they're starting to invade now. They let go and they start to crawl. But remember this, a tissue isn't just a bunch of cells sitting together. A tissue is cells locked in by proteins all around, the extracellular matrix. Everything is like held together. Otherwise, you would just fall apart into cells anytime you sneeze. So if a cell wants to move, it's going to be hitting extracellular matrix. It can't really get anywhere. And also to invade into the mesenchymal area, I said earlier, you've got that basal lamina, which is a really thick protein layer. If cells are going to invade, they need to get through that protein. How do they do that? Thank you for asking. They do that by releasing plasminogen activator. Plasminogen activator. Okay, there's cell crawling again. All right, plasminogen activator. Okay, so PA is an enzyme. You release it, and it will act on plasminogen. Plasminogen. Gen is to make. So plasminogen. So this is something that's going to make plasmin. So plasminogen activator will activate plasminogen, which then becomes plasmin. Plasmin is also an enzyme, and it will activate matrix metalloproteinases. So these are proteinases that are on the extracellular matrix. They're sitting there inactive, just ready to go when they're needed. So they're sitting there inactive, and the plasmin goes in and activates them. Now you've got all these proteases. They start chewing up the protein, and now the cell's got a path that it can go through. So the three things necessary for invasion and metastasis are let go, crawl, clear a path. And you're doing that by, my daughter's teasing me. You're doing that by releasing your focal adhesions and your adherens junctions, activating RAC-RO CEC42 to start to crawl, and releasing plasminogen activator to activate those matrix metalloproteinases and clear a path. Bum, 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 bum. And let me see, I think if I'm not incorrect, That is your 30-minute lecture for today. That's about all I have. See, normally, I'm walking around, I'm talking my stuff, I'm wasting time. Everyone's writing stuff down. I'm writing stuff up. These lectures take a long time. That was an hour and 15-minute lecture that I just gave you in 30 minutes. So there may be some info I forgot too, because I don't have my notes with me and I'm just kind of coming off the top of my head. This is what, 26th, 27th time I've been doing these lectures. So I remember a lot of it. Not all of it. Okay, any questions, comments? I am going to let you go if you don't have a question. Oh, here's our plasminogen activator. Here's your basal lamina. If these cells want to get through, they need to chew through that. And cigarettes. Anyone need a cigarette? Nobody needs a cigarette. No smoking in my class, God damn it. Okay, here we go. Why does heart not count? When do we have exam 3 is next third. We already had exam 3. Exam 4, Dr. Quach, will be next Thursday. Don't be sorry, just get it right. Sorry, Daniel. I'm just being a jerk. See, I can't yell at students when I'm at home. I can yell at my kids, but they don't care. Any questions or comments? Any discussion? Is this everybody? We don't have many people today. I still keep your sunglasses. Good, good. They're designer sunglasses. They're the good ones. Edgeman, how you doing, girl? Give me a thumbs up. Let me know you're paying attention. Come on. Yes. There's the Edgeman. Okay, I got nothing. I'm just wasting time now. Anybody have any questions or comments or what? Is that the end of the chapter? Nope. One more lecture. We've got, in the next lecture, we'll talk about the molecular aspects of cancer. So this was kind of like the bigger, broader section. Now, then we'll go into, you did a good job covering everything. Well, thank you, Jenna. Thank you, Jenna. It's good to get a little positive feedback every now and then. Mostly, I'm around the house with my wife, and all I do is get yelled at here. Okay, are we done? Shall we stop? Thank you, Adelina. I will have a good one.
Have an excellent day, Dr. Slish.
Thanks. You too, Raphael. You're a good man. All right, I'm shutting her down.